Viroid, Virusoid and Prions. These are all the plant and animal pathogens which causes diseases in plants as well as animals and humans. Now we'll see their general characters, what are all the different types of disease caused by the each and every pathogen. And my name is Janet Shankar Reddy and I'm doing PhD Plant Pathology in Annamalai University. I'll say starts with the viroids. Viroids are small, covalently closed circular single-stranded RNA molecules. It contains no protein coat, which means they are small, covalently closed single-stranded RNA molecule, whereas virus genome is made up of DNA or RNA, but not both. But here, all viroid genome is made up of RNA only, whereas viruses contains protein coat. Here the main difference between virus and viroid is virus contains protein coat, whereas viroid is devoid of protein coat, which means contains no protein coat. It was first reported by Diner and Raymer is the scientist who reported viroid. Diner 1971. The first reported viroid is potato spindle tuber viroid. Potato spindle tuber viroid in the year of 1971. This is the potato spindle tuber viroid symptoms. You can see this is the normal potato. The abnormal, shapes are like small protuberances can be observed here. So they worked on it and finally discovered a new type of pathogen that can cause infection in plants, that is viroid. Coming to the discovery, there is a this is a small timeline which starts in 1960, Raymer with O'Brien, and then it gone on in 1971 where the Diner first demonstrated about this viroid. In the year of 1978, first viroid to be sequenced, that is potato spindle tuber viroid in the year of 1978. This is a timeline for viroid. Now we'll see what are all the general properties of viroids. See, one more thing that we have to observe here, virus disease can be reported in plants as well as humans, but there is no viroid disease has been reported so far in humans. Viroids are only meant for plants only. So now we'll see the general properties. They are usually smaller than that of viruses. So these viroids are smaller than that of viroids, which is known to infect by the plants. So genome size ranges from 250 to 401 nucleotide longs, which have the high degree of specificity. So each and every viroid has their own specificity from other viroids. So here one more thing is that it contains no protein code and it do not code any code proteins. It do not code their own code any code proteins. So they replicate individually into the host plant associated with the plant virus for their replication and their functioning. Here how this viroid can replicate, it can depends on the host plant or with associated with the plant virus. So these viroids can associated with the plant viruses, and a few of the times it will depend on the host plant for their replication and other functions. So viroids are only transmitted by mechanical or sap transmission. But before that, what we have seen, viruses can be transmitted by different types of agents. That may be sap transmission, that may be grafting, that may be seed materials, that may be fungi, that may be insect vectors. So many are there. But here the specialty is viroids are only transmitted through mechanical transmission or sap transmission. So far, no vector transmission has been reported. So far 40 viral disease has been reported that causes a plant disease in the world, only 40 viral disease, but so far no more than 2000 plant viral disease has been reported, but viroids are only 40 has been reported so far. And whereas the virus moves from cell to cell through plasmodesmata, the viroid also moves from cell to cell through plasmodesmata and cell divisions. So during cell divisions, the plant viroids are also transmitted from one place to another place or one cell to another cells. And the replication mechanism of a viroid include a rolling circle mechanism. Replication mechanism include rolling circle mechanism, whereas the virus has used the host cell machinery to produce the particular protein that the virus requires. So the viroid disease, we will see viroid disease, that is the most important one, or the first discovered viroid is potato spindle tuber viroid. We can see the abnormal symptoms coming to small protuberances, abnormal shapes, small in size. These are the different types of symptoms that is produced by the viroid disease, that is especially potato spindle tuber viroid. The second one is citrus exocardosis viroid. Now symptoms include shelling of bark. We can see here the bark is shelled here also. Yield loss, automatically yield loss transmitted through stalk and graft. So this viroid is transmitted through shock and so one of the other important viroid is coconut kadang kadang viroid. Here the size can be reduced and the husk production also can be reduced due to this coconut kadang kadang viroid. So tree length can be reduced and stunted. Tree length can be reduced and stunted. So the most diagnostic and prominent symptom and important symptom that can be observed for coconut kadang kadang viroid is withering of leaves and the infected plant look like a telephone like appearance. The infected plant look like telephone like appearance. We can see here the infected plants, the leaves all get withered. There is no leaves left here, and this plant look like telephone like appearance. That is the most prominent and diagnostic symptom of coconut kadang kadang viroid. This is also the symptoms we can see here. Reduce the size of plant can be reduced. You can see here abnormal withering of leaves. The leaves get withered, tadpole like appearance. So these are different types of yellow color discolorations, like small stripes can be seen here. So these are all the general symptoms of coconut kadang kadang viroid. Now we will see about virusoid. So virusoid is a circular single-stranded RNA dependent on plant viruses for replication and encapsulation. So here, virus requires no other viruses for their multiplication, all those things. But viroid requires few of the times it may requires viruses, or it can dependent on host cell machinery. But virusoid here, it is also single-stranded RNA which dependent or which required on plant viruses for replication and encapsulation. For replication and encapsulation, the virusoid requires plant viruses. It was first discovered by Randless, the scientist who discovered virusoids. This is the structure of a virusoid. We can see here different types of it's almost look like a protein structure, maybe a malfunctioned or changed protein. So it contains several hundred nucleotides. It also does not code any code protein. So the proteins which are present in virusoid do not code any code proteins. So almost similar to viroids for their replication mechanism. Here viroids are replicated by means of rolling circle mechanism, whereas the virusoids also replicate by means of rolling circle mechanism. Almost same in replication, but there is a slight changes here. So virusoids act as a satellite virus and not necessary for viral RNA. So few of the times what will happen here, the virusoids act as a satellite RNA. So satellite RNA means which need another virus for the infection. So they act as a helper virus for the to other viruses for sometimes for infection. So the molecular weight ranges from 1.5 to 10 to the power of 6 Dalton. So the next one is prions. So here the prion is a small infectious particle composed of abnormally folded protein. Abnormally folded protein. It's not a pathogen. It is a malfunctioned protein. It is a malfunctioned protein. So this is composed of folded protein and has the ability to transmit their misfold shapes into the normal variants. So once the very simple, it is a misfold or it is a malfunctioned protein or misfolded protein, and it has the ability to transmit their characters into normal variants or the normal proteins. So it was first discovered by Stanley B. Prusner, is the scientist who discovered prions, and he also named as prions. So for the discovery of prions and prion diseases, Stanley B. Prusner received Nobel Prize in Physiology and Medicine. Now we will see the different types of prion disease which is caused by prions. In sheep and goat it causes scrappy disease. Scrappy disease, we can see here skin get slightly loosed off, and it almost starts to behave like abnormally and look like a little bit weak than usual plant. This is scrappy disease. What will happen after infection of 6 to 8 months? The animal will die, and uh, so the report of this disease in animals is getting increased year by year. So in sheep and goat scrap, in cattle, important one, in cattle it called mad cow disease. Mad cow disease, that is bovine spongiosperm encephalopathy, that is mad cow disease. So the infected cow or infected a cattle, what happened here, they lost control, they can't able to stand on their own legs. So after infection of a few weeks, it start to behave abnormally, and completely lost control and standing in animals. In humans also, few of the diseases reported by prions, that is especially Jacob disease as well as Kuru disease. So very important diseases. And due to Jacob disease, what will happen? The brain will start to send the signal to different body parts. So due to the body parts can't able to coordinate or control their by own, we can see the person who are infected by the crude disease. What will happen here? He completely lost control. He can't able to stand there by own. So within the 14 months, the man or the person who are get infected with this prion crude disease, he will die. So before that, the affection of limbs, signal transmission from brain can be switched off, you know and the body parts, we can't able to coordinate the body parts. We can see here the different types of symptoms in this particular man. And also nutrient absorption can be reduced. You can see how weak he is. You can see the ribs here, how weak he is. So these are the different types of symptoms which is produced by the prions.